Very good morning to one and all present here. My name is Dr. P. Samuel, Assistant Professor, Department of Biotechnology, Ayanada Janaki Ammal College, Sivagasi. First, I wish to convey my thanks and regards to the management of this college and most respected principal for giving me this opportunity to prepare the video lesson for the welfare of our students. The topic of the lesson is in vitro fertilization. Again, the topic of the video lesson is in vitro fertilization. You may ask questions there, what is fertilization? The answer is fusion of gametes. Fusion of gamete is called fertilization. Gamete in the sense male and female reproductive cells. Male reproductive cell is called as sperm and female reproductive cell is called as ovum. Fusion of the male, sperm and ovum is said to be fertilization process. Usually this process takes place in the fallopian tube, but the name of the technology is in vitro fertilization. What is in vitro? In vitro refers to performing an experiment within the laboratory glassware. I repeat, performing an experiment within a laboratory glassware is said to be in vitro. Antonym to this term is in vivo, which means performing the experiment directly within an animal model. So, as the name suggests, in vitro fertilization refers to performing the experiment in the laboratory. So, what is the procedure all about? Ultimately, it is the advancement of medical science which help us to fuse the male and female gamete within the laboratory glassware, and this technique will help the people suffering from infertility problems. So let's move on to the topic. The first step in the in vitro fertilization is the collection of sperm. I repeat, the first step in the in vitro fertilization procedure is collection of sperm. See, for an healthy adult, the total number of sperm in the semen is in the numbers of millions, whereas in a female, usually only one egg will be produced per month. Think about the correlation. The target is 1. Millions and millions of cells are moving towards the target. So once a veterinary university professor said, reproduction is a luxurious process. And this word attracted me a lot, luxurious. So I started to study various papers. One of the paper in the leading journal called PLOS ONE says, an average healthy adult can invest 101 kilocalories of energy during the process of reproduction, whereas a female, she invests around 69 kilocalories of energy. So I concluded that the statement said by the professor was literally true. It is a luxurious process. So during the process of reproduction, the semen will be ejaculated into the reproductive canal. I'm talking about the natural process. The sperm travel from the genital tract into the fallopian tube in search of the egg. So this will happen during the time of natural process of reproduction. In the case of laboratory procedure, the individual is subjected to artificial ejaculation procedure, so that the semen sample will be collected. The collected semen sample will be examined under microscopy for the morphology of sperm, as well as it is analyzed for any disorder associated with sperms. There is a condition called oospermia, in which no sperm cell will be present in the semen. So this is the most common problem associated with the infertility problem nowadays. Apart from this, there are many other disorders associated with infertility problems, especially from the human male. So, after the semen is collected, it is microscopically examined to distinguish healthy and unhealthy sperms. So, the healthy sperms are collected and they are stored. And the partner for the sperm is egg cell. It should be retrieved from the human female. I wish to recollect that healthy women could produce only 1 egg per month. So, on the same day itself, the egg also have to be collected from the female. So, there are drugs which enable us to hyperovulate the female. One such drug is called Clomid. This will be given as 50 milligram per day for 5 days, so that multiple eggs will be produced. From the follicles, the egg is also collected with the help of ultrasound guided procedure. The egg is also collected. The collected egg is examined under microscopy to analyze the healthy and quality of the egg. Right. After collecting the two cells, the sperm cell and egg cell, it is allowed to fuse in the laboratory condition. It is achieved by mixing both male reproductive cell and female reproductive cell in the laboratory glassware, which enable the egg to fertilize, which means the meeting of sperm and egg cell will happen in the glassware itself. The fertilized product is said to be zygote. Later, the zygote is transferred into the female structure called uterus, so within which the zygote will develop into baby. And by words it seems to be very simple, but it is a laborious process and it is a costly process also, because there are a lot of things to be considered while examining both sperm as well as ovum. In case of ovum, there are various disorders. Some sperms are lagging in their motility. Some sperms showing different structural deformities associated with the cell. So, a typical lab technician with the guidance of a physician is very essential for choosing the healthy sperm for the procedure. So, after considering all these things, as I told you before, the eggs will be fused with sperm in the laboratory itself. The fused product called zygote will be transferred into the uterus. There are two other forms of assisted reproductive technology. They are called as ZIFT and GIFT. ZIFT refers to zygote intrafallopian transfer. GIFT refers to gamete intrafallopian transfer. Both GIFT and ZIFT are different from in vitro fertilization, because in GIFT, in gamete intrafallopian transfer, the gametes are directly transferred into the fallopian tube. Whereas in IVF, the fusion is taking place within the laboratory glassware. Right. In some critical conditions, there is a problem called ectopic pregnancy, which means the baby started to develop within the fallopian tube itself. Early detection of the malformation of the baby in the fallopian tube might help the doctor to save the mother. If the baby started to develop within the fallopian tube, gradually the tube will burst. It eventually led to very critical condition of both the baby as well as the mother. So there are problems associated with fallopian tube. There are problems associated with the sperm motility. There are problems associated with egg formation. To overcome all these problems, medical assistance is required. So, with the advent of technology in the field of medical science, one such procedure is called in vitro fertilization, which help the couples suffering from unexplained infertility problems. And as I told you before, it is a costly process which takes around 1,30,000 rupees as per the Indian price, but the success rate is around 85 to 90%. Right. So once the zygote is implanted into the uterus, it started to develop into a baby. There are different stages in the development of embryo, and the study of the developmental stages of an embryo is said to be embryology. I am not going into embryology topic. Once the egg is implanted into the uterus, after 5 to 7 days, a structure will be produced and it is called blastocyst. So blastocyst is a hollow ball of cells which will be formed after 5 to 7 days of zygote formation. And this blastocyst is rich in a super type of cell called stem cells, and this stem cell is called the embryonic stem cell, and it is pluripotent in nature. We will discuss what is pluripotent later. The early stage of life, every life starts from the single cell called the zygote. The hollow ball of cells contain a mass of cells said to be embryonic stem cell. They will be harvested, preserved and applied for various medical problems. See, most of the people would protest this procedure because harvesting the blastocyst is equivalent to killing an individual. But scientist says the life actually begins after 14th day of egg implantation in the uterus. But we are harvesting the cell during the 5th day. So it is not actually killing an individual, but it is equivalent to treating a cell. I just want to make this point very clear. Harvesting blastocyst is not equivalent to killing an individual. So this is the major problem raised by public in stem cell research. Fine. After harvesting the blastocyst, the cells will be removed. Because human blastocyst, we are triploblastic animals. The blastocyst consists of triple layers. So the inner core contains the embryonic stem cell. They will be harvested. Harvested cells are inoculated into the media. So media is the nutrient supplements which enable the cells to grow and proliferate and establish. By inoculating the cells into the suitable medium, the stem cells will grow, they will flourish, they will establish. And this is the simplest procedure for harvesting the embryonic stem cell. The opposite one is called adult stem cell. Stem cells can also be harvested from adult individuals. This is called as adult stem cell. Examples are bone marrow cells, a typical example for adult stem cell. See, we are all familiar with the structure of bones. So within the inner core of bone contain a brownish material, a reddish brown material, and the reddish brown material is called bone marrow. Scientists used to say this bone marrow as the base of all cells. So it is the foundation from which all other cells will develop. So this is exploited during various medical procedures. And once the cells are harvested, once the stem cells are harvested, it is very important to preserve them. The process is called cryopreservation. There are many cryopreservation banks in India. One such bank is Live Cell in Tamil Nadu. What they are doing is they are harvesting the cells. The harvested cells are subjected to cryopreservation, or otherwise called as short freezing, where the cells are preserved under minus 196 degrees centigrade. The process is otherwise called as lyophilization. This will be carried out in the liquid nitrogen condition, which help us to preserve the cells under the sub-zero level. You may ask questions, sir. Below 0 degree, ice crystals will be formed. This may harm the cells. But while preservation, the medium and cell mixture is treated with the cryoprotectants like glycerol, sucrose, trehalose, which protect the cells from being harmed by the ice crystals formed during lyophilization process. A recent article says stem cell preservation is a very important one and also a novel procedure which prevent a baby from more than 85 critical medical conditions. So obviously it is a recommended procedures by the doctor to protect the baby from critical medical conditions. So I want to recollect the process called the cryopreservation, which means preserving the cells under sub-zero level, particularly minus 196 degrees centigrade. After cryopreservation, the cells can be taken for, or it can be applied for treating critical medical disorders. One such disorder is called heart failure. Heart failure is a major problem in India. So, for treating the procedure, we used to follow some invasive procedure. Doing operations, some critical condition, it may require heart transplantation also. But stem cell is a boon to treat such critical condition because stem cells are with a property called homing, H-O-M-I-N-G. Homing enable the stem cell to move directly into the affected area. So like injecting a vaccine or like injecting an antibiotic, the stem cell suspension can also be injected through the vein. The vein will collect the cells into the heart where the cells are implanted for a few minutes for proliferation and differentiation. Sir, you may ask a question. Sir, heart is not a static organ, rather it is a dynamic organ, which means it will pump every time. So what is the procedure required to stop the cell, or what is the procedure required to prevent the stem cell from moving into other areas during the procedure. So a scientist from Japan, she suggested the stem cells are coated with ferromagnetic nanoparticles, which help us to maintain, or which help us to provide a static condition for the cells for a few minutes. So within the short span of time, the stem cells could anchor into the tissue and develop into a matured cells. The stem cells are rightly said to be a blank cell. Actually, they are not given with any function at the time of its production. You may be wonder, sir, what is blank? We used to refer, I am blank, feeling blank in sometimes, because we don't know what to do at sometimes. Likewise, the cells are also in the blank stage. Upon receiving the signal given by the other cells, the stem cell undergo another process called differentiation, which enable the blank cell into a functional cell. If the signal arises from the heart, it is differentiated into cardiomyocytes. From glands, it started to produce hormones. The signals are probably from our own side itself. The signals alone make the stem cell to differentiate. Upon differentiation, a stem cell could behave as a functionally active cell. This functionally active cell will reach the spot and do magics within the damaged area, because there are critical conditions associated with organ transplantation itself. Because organ transplantation, it is a sequential clinical procedure. So before transplanting an organ, we have to check two important things. One is called blood group of the donor and blood group of the recipient. It should be matched in a matched condition. Apart from blood group matching, there is another criteria called the HLA typing. Even the condition of satisfying both blood group as well as tissue typing, it normally takes 6 more months for the transplantation procedure, invasive transplantation procedure. So before the 6 months time period, whatever may happen to the recipient, if the unmatched organ is transplanted into the recipient, could lead to severe condition called graft versus host rejection. It is a serious immunological problem. So the stem cell transplantation is a non-invasive procedure. It doesn't require any laborious operation theatre procedures and all. A minor hole will be drilled in the recipient. So through the drill, like injecting a vaccine or like injecting an antibiotic, the stem cell suspension is injected into the recipient. So as I told you before, the stem cells are confined with a property called homing. This property leads the stem cell to reach the correct area, the damaged area, where they settle. They proliferate, they differentiate and started to behave as a normal functional cell. See, in developed countries, with the consent letter from the people appearing for IVF, the unused embryos are used by the doctors for stem cell research. So, what actually required is the legal consent letter. So without the letter, we cannot do any research in the embryos because it is scientifically not ethical one. After getting the consent from the couples, the unused embryos are used for the production and application of stem cells in critical diseased individuals. Guys, this is all about IVF and the fate of unused embryos in stem cell technology. Thanks for watching our video. Stay connected with us. Subscribe our Untitled Digital channel. And don't forget to click the bell icon to get the latest updates. See you in the next video. Thank you.